Yeah. So, so. This is going to be the last revision stream before exams. What I found recently when doing these revision streams is they are working for doing things like big mind maps, kind of just putting a bunch of information on the board. But what they aren't working for is exam questions, because what I do is I don't know why, but when I stream, I tend to answer the questions very differently to how I would when I'm sat in silence writing. So I'm not doing any exam questions on stream. Instead, I'm going to spend the next hour just doing some general revision. And then what I'm going to do is I'm going to sit down and do a whole past paper from 2023, use a mark scheme to answer it. And then I'll do a stream later where I go and review all of the things that I need to work on. So if I notice, for example, when I do the past paper in an hour, if I notice that I really struggled at the section on gas exchange, then we'll spend another hour doing gas exchange. If I struggled on a lot of stuff, we'll focus on a lot of stuff. So 1 hour of unfocused revision just for everything, then an hour of past paper revision, and then an hour of focused revision. That's the deal today. Now, before we get into things, some very, very exciting news. My plants. So, basically, for those who don't know, I've been my dad is a gardener, and what we decided this year was to just grow a bunch of stuff. So what we now have, five peppers, 5 chili peppers. Great. I might need to go for a second because hoovering. So we now have 5 chili peppers, 2 cucumber plants. We have, I'm pretty sure it's like a butternut squash. We have a load of tomatoes, a load of strawberries. And yeah, I'm genuinely ecstatic to be able to watch these things grow over the summer. It's going to be a lot of fun. The problem is one of the plants might be dead. One of the cucumber plants is kind of beginning to fall off a little. I ended up accidentally snapping like the very bottom of the stem when I was trying to like push it in slightly because it was really wobbly. And I can't tell whether I've killed it or not because it seems to be fine like a couple of days after. It's just incredibly floppy now to the point where like it'll just do this, you have to balance it on something. So I don't know if I've killed it or just paralyzed it, but yeah, we have a load of plans. It's a lot of fun. So I'm thinking, what do we revise today? Because in school, we've just gone through infection and response. We've completely gone through that. We've completely gone through a lot of the generic cell things. So I found out the cells, the topic cells, takes up the most space in exams, typically. So that's things like cell fractionation, the structure of prokaryotes and eukaryotes, I guess DNA, that takes up the most space in an exam. So logically speaking, that's the topic we should focus the most on. I'm just trying to think, like, is there any other topic? We could quickly go through DNA replication and transcription and translation. We could very quickly go through that. Because this is a topic which I often am unable to explain to a certain degree. So what you have is, let me see if I can remember the structure. Your phosphate group, is it I think it's a ribose sugar, and your nitrogenous base. That's the structure of a nucleotide. And then from there, it just repeats because that's what, that's how it works. Now, the interesting thing to note is there are two different types of bonds. Two different types of bonds in nucleotides. You have the phosphodiester bond, or I'm just going to put the PD bond, and the hydrogen bonds, which join, these join adjacent nucleotides, those join bases, complementary bases. So, the way the bonding of this works is, I'm just going to draw like a very zoomed out version here. So, imagine that the red here represents the hydrogen bonds. Okay. So what's first going to happen is, it is DNA, is it DNA polymerase? Firstly, DNA polymerase is going to, basically, I don't know whether it hydrolyzes or not. The thing is, mark schemes are really picky with the wording you use for this kind of process. They are very picky. Like, I think you can drop marks for mentioning hydrolysis. I think you can lose marks for mentioning DNA polymerase, even. So you lose a lot of marks for putting things which are right. So I'm just going to put that they hydrolyze the hydrogen bonds in between the bases, which causes it to unzip. Like so. It causes it to unzip because you've broken all of the hydrogen bonds between the two strands of DNA. Now, what happens from here is, if we talk about DNA replication, the thing that happens is, on each strand, you have your free-floating DNA nucleotides, which are gonna join to their complementary bases. So, A will join to T, etc. So your free-floating bases will now join up to their complementary sides, and this happens for both sides, both strands of the DNA. Like so. And then by the action of DNA, is it DNA synthase? DNA... Okay, I made a mistake. That's DNA helicase that breaks the bonds. That's DNA polymerase. DNA polymerase joins up the phosphodiester bonds between these new nucleotide strands in order to then give you your two new strands, and that's called semi-conservative replication. I don't know whether something rejoins the hydrogen bonds or whether the hydrogen bonds just form again between the complementary. I'm honestly not too sure. So that might be something to look at. But there you go, you have your two strands. In fact, I'm pretty sure there was an exam question, but I have over there about it, so I can quickly check. But anyways, in RNA synthesis, what you have is, your DNA will split, your DNA will, unzip, but only one bond is used in the replication. And what you have, again, is your free-floating nucleotides will bind to their complementary bases, And then by action of RNA synthesis, it creates this smaller copy, which then goes on to be used in protein synthesis. So that's why, in an exam question telling you to describe differences between DNA and RNA, that's why you're allowed to say that it's single-stranded, it's a smaller copy. Obviously, in RNA, in DNA, it's ATCG, but it's AUCG in RNA. Obviously, the U being uracil, not thymine. And also the whole thing of introns and extrons. So introns are non-coding DNA, which is removed during splicing. At this stage, it is pre-mRNA. Meanwhile, after splicing, after it leaves the nucleus properly, that's when it then becomes RNA. In prokaryote DNA, it does not have introns. It does not have introns. Let's also very quickly go through a couple definitions, some of which I didn't even know until a couple of days ago. So here we go. DNA is degenerate. Like pyrocynical. It's a pyrocynical word, okay? What that basically means is that multiple codons, remember, in questions where it asks you about the base sequence, always refer to triplets. Basically, every multiple codons code for same amino acid. Right? Tasty codons. But I don't know if they're tasty. How you doing, man? This is a last revision stream before I'm basically in an hour gonna do a past paper. So it's one I've never seen before, and it's one that no one else can access. I literally had to ask Miss Collinson to print it out because it's a 2023 paper. I'm doing good. We just went to the garden centre and I bought a load of peppers. I bought the spiciest pepper in the world, and I bought 2 relatively tame ones. So I now have a whole arsenal of peppers. Yeah, I'm not showing it on stream. I don't know if I legally can. But, so I bought a whole arsenal of peppers. I think I killed one of the cucumber plants by snapping the stem at the very bottom. I hope I didn't, but... And, yeah, we bought a load of tomatoes. So we literally have, like, 10 tomatoes. It's not that one. It's the Scotch bonnet, which I think is in the 100,000 to 150,000 range. So it's still very spicy, but not as spicy. Yeah, I was trying to plant it like further in, but I ended up snapping it. It's very flimsy now, the cucumber plant. I think I killed it, but we're gonna put them in the grow bags. We're gonna put them in when it's warmer, so maybe a couple more days. It's meant to be 20 degrees in a couple of days, so we'll do it then. And then, yeah, the growing will begin. My 5 peppers, my 2 cucumbers. We have about 10 tomato plants, 10 strawberry plants. I think we also got, I can't remember what you call it. It might be a butternut squash. We just got that because it was funny. So we have all sorts. But anyways, we're gonna keep revising for about 30 minutes, and then I'll do a past paper off stream, and then later I'll do a stream where I kind of evaluate what I need to work on. Because remember, guys, the biology exam is in less than a week now. It's officially less than a week until the exam. So we gotta work. So anyways, degenerate basically means that multiple codons code for the same amino acid. And you can use codon tables to kind of verify this. For example, I'm just making this up, a sequence of A, AT, AT, and ATAA, maybe that codes for the same amino acid. And what that means is during mutations, DNA won't always code for a different amino acid. It won't always mess up the protein structure. In an exam, if you get asked how a mutation can cause a change in the protein structure, refer to the fact that the triplet sequence has been changed. That's like a key thing. The whole idea that it's a base sequence of DNA in triplets that affects the sequence of amino acids. That's like the key definition right there. That's how you get 3 marks in an exam. You say that the base sequence of DNA in triplets affects the sequence of amino acids in the primary protein structure. Another definition I didn't know is universal. All this means is that all organisms in the world have A, T, C, and G bases. They all have the same nitrogenous bases. And the final thing is, I think it's called non-overlapping. All this means is that during translation of DNA, each base is read once. Each base is read once. So DNA doesn't overlap. I'm pretty sure that's all that means. So that's kind of everything for DNA there. That's kind of everything. I'm not sure if I've missed much or not. But again, mutations, obviously you can liken the likelihood of the mutation through mutagens. But yeah, let's keep it going. Let's talk about adenosine triphosphate. ATP is like my favorite molecule. It's very fun. And you need to be able to talk about the 2 functions of ATP. And to be honest, I didn't even know that this second function existed. I didn't even know. Oh. Yeah, I did not even know about this function. So you have adenosine triphosphate. So you have your base adenosine, or adenine, sorry. You have your sugar, your ribose sugar, and then you have your 3 phosphate molecules. Now, in ADP, this high energy covalent bond is broke, which creates ADP and releases an inorganic phosphate, PI. Now, this phosphate is actually used for more than one thing. So, number one, the energy released during this reaction, during the hydrolysis of ATP, can be used for a number of reactions or named processes. For example, active transport, protein synthesis, things like that. But this phosphate is used for phosphorylation. I don't know if I've spelled that right. Phosphorylation. And what this basically means is that this inorganic phosphate is able to alter the structure. It's able to change the shape of, I guess it would be like a molecule. It's able to increase the reactivity of a molecule or a substance. I don't know what the word would be. It's able, hold on. Because it's a word that I completely forget. It's able to alter the shape of something and change the alter the shape. Two ways the hydrolysis of ATP is used in cells. Here we go. This was a question that I lost both marks on. Basically, during two ways hydrolysis is used in cells is provides energy for. It provides energy for substances. Or provides energy for reactions. So there you go. It provides energy for reactions, and the phosphorylation of other substances causes them to change shape. Change shape, or basically become more reactive. That's what phosphorylation is. It changes the shape of a substance / makes a substance more reactive. Let's look at some more improvements from this paper here that I need to work on. A big important thing is inferring things from graphs. I can't do this for the life of me. So some little tips that I picked up really is, firstly, let's look at this graph. It's basically how the rate of hydrolysis of sucrose increases as these plant cells develop. It talks about it all in like this statement thing. But anyways, what you really need to know is what can you conclude about the growth of plant cells from the data. So look, to begin with, there is barely any hydrolysis of sucrose. But as time goes on, it increases and then eventually stops. It flattens out. It reaches max. So what you need to say there is the plant cell, what I put, was the plant cell's growth rises because there is a higher hydrolysis, which means more glucose. I rambled there. What I really had to say was sucrose hydrolysis, which is the graph we're measuring, is linked to some growth. That's all I needed to put for a mark. Literally saying, it says there, what can you conclude about the growth of plant cells? I can conclude that the growth of plant cells may be linked to the rate of hydrolysis of sucrose. Literally just how is that linked to the y-axis? And you get a mark for it. Kind of mad. Anyways, what I'm going to do now is I'm going to actually do that past paper. I am meant to be revising for another hour, but you know what? I'm going to do the past paper now. I'm going to sit for an hour and do it. Maybe have a break in between to eat. I'll sit for an hour, do it, see how much I get through. And then what I'm going to do is I'll go back on this stream and yeah. Alright, so for you guys, that would have been an instantaneous cut. For me, that was an hour and a half of back-breaking revision for the grand conclusion. So, I didn't do all of the paper. I missed out on the final reading question, and I missed out on a lot of question 8. So, I missed out on 24 marks that I would have otherwise maybe gotten, maybe not. So this is out of 51, the paper. And out of 51, I got 35 correct. So that is 35 out of 51, or about 68%. I would argue that that is a pretty good percentage for paper 1. Bear in mind, I had seen actually 2 of these questions, the first 2 here, which I'll get onto in just a sec. Bearing in mind that I think I did very well, considering there were a couple of those questions which, looking back, I really could have easily gotten. I really could have easily gotten some of these questions right, but I didn't. So let's go through it. Number 1. Describe the primary structure of all proteins. I got this right, 2 out of 2. I said it's a sequence of amino acids joined together by peptide bonds. And that was exactly what the mark scheme was after. So 2 very nice marks. The next question is describe universal, non-overlapping, and degenerate. I got the definitions wrong. I spread misinformation. I actually got these definitions wrong. Universal means that the same codons always code for the same amino acids. So basically, if we had a sequence ATAA in one organism and the same sequence in another, no matter what, it's still coding for the same amino acid. That's all that means. So the same codon always codes for the same amino acid. Non-overlapping just means that each base is only part of one codon. So for example, this and this wouldn't happen, basically. Each base is only part of one codon. So those are some definitions to work on. So I missed 2 very nice marks there, purely because I made a mistake. Degenerate is multiple codons code for each amino acid. Next, describe how the appearance of the area containing DNA would differ in a prokaryote to a eukaryote. I made a mistake here. I started talking about chromosomes and how in prokaryote it's free-floating and supercoiled. Not what it was asking for. It was asking how the appearance of the area is different. So I would have had to talk about the nucleus and then the nucleolus. That's all it was asking. So an easy mark dropped. I dropped some pretty bad mistakes in a question which I thought I did really well on. Describe the mechanism of breathing that causes air to enter the lungs. What it was asking for was the diaphragm. The diaphragm flattens out as the diaphragm muscles contract. So basically, the diaphragm muscles are contracting. This leads to the diaphragm flattening out. Okay? And for a second mark, the external intercostal muscles contract, which leads to the rib cage pulling out, or being pulled out. Again, 2 very easy marks there, which I will definitely remember now. Literally, it's the pillar metaphor that I was using before. It contracts, which causes it contracts, which causes it to flatten out like a pillar. That's literally, and I missed out on the flattening out bit, which is kind of silly to be honest, but hey. And then yeah, I didn't really know this. I don't think I've ever written about the rib cage before, but it makes sense. When you breathe out, when you breathe in, you can feel your rib cage pushing up against your skin, I guess. You can feel your rib cage push out. So yeah, the rib cage is pulled out, the diaphragm flattens out. There you go, out. That's literally it. When you breathe in, The diaphragm flattens out, the rib cage is pulled out. Next was a suggest question. I always suck balls at these questions. Suggest questions, because I overthink them. I overthink the suggest questions a lot. It was basically wanting you to talk about how a thicker slime capsule means it's less likely for a phagocyte to bind to the cell wall because the capsule is thicker. I talked about how that increases the width needed to permeate the walls. No, it wasn't asking for that. So I got a mark. Next is draw a box around the R group of a fatty acid. Basically, that is the only part which is not the R group. All of that is the R group, and I didn't know that. But now, kind of looking at it now, it makes sense, because it's a fatty acid, so it's obviously going to have your COOH, but everything else is going to be unique to it. So yeah, simple mistake. I got the bit about describing. I'm good at that question. This contrasts the structure between triglycerides, phospholipids. Explain why phospholipids can form a bilayer, but triglycerides cannot. I'll be honest, I cheated this question. I googled it, and I still got it wrong. What it was asking for is the phospholipid has a hydrophobic head, hydrophilic chains. Triglycerides only have hydrophobic components. What this means is in a bilayer, it causes the bilayer structure to form because the hydrophilic point outwards, the hydrophobic point away from the fluids. Basically, triglycerides, triglycerides are only hydrophobic. So here, this is all hydrophobic, but in this, that's hydrophilic, that's hydrophobic. Also, a big important thing which I forgot is in unsaturated, the tail of a fatty acid kinks. It's slightly like lopsided, and I didn't put that, but that's a very important fact. So far, I need to work on explaining inspiration and expiration. I need to work on a couple definitions for bases and DNA, and I need to work on my definition. Well, I need to work on my general knowledge of lipids. Next was talking about, literally this question here, talking about why the fluidity of membranes would be higher after a load of unsaturated fatty acids are put on the thing. It's because unsaturated. And also, I'm really pissed off about this. You would have gotten 2 marks for literally just stating what happens, right? Less of A, more of B for a mark. That means that fluidity is linked to an increase in fatty acids. Bruh, I always miss that. In exam questions. I always miss how something links. How it links to the topic. I always miss that. In every exam question I've ever done relating to suggest why, explain how this links. I never explain how it links. It's a genuine mistake that I always make. The next question was a math question, which I did pretty well at. I almost got it wrong, but then I realized how you did it. One feature that shows the images were taken with an electron microscope, not an optical. You would have had to give a named example there, and I didn't, but I think I still would have gotten both marks. State a role of a helper T cell. I put activate cytotoxic T cells, correct. Here's something, translation. In translation, the tRNA brings a specific amino acid by binding to the complementary codon. The anti-codon binds to the anticodon. And I put brings complementary amino acid to ribosome by binding anticodon to codon. I would have gotten that. Next was a nice surface area to volume ratio calculation. The next one, I really just misread this. It said, after collecting the data, the student noticed that shape C block 2 was damaged. Now, I'll be real, I read this as shape C being damaged. So I just assumed all of this was wrong. What it really wanted you to do was just disregard the data there, which I didn't, because I misread the question. So yeah, I misread the question, and I dropped a mark, and I almost dropped another mark just for the misreading. In this one, state 3 variables the student controlled. Apparently, mentioning volume wouldn't have gotten you a mark, so I dropped a mark there. I did well at just explaining how surface area to volume actually works. I got the definitions of DNA helicase and polymerase wrong, okay? And I'm actually very shocked at this. So DNA polymerase basically breaks, literally all it does is breaks H bonds between bases. That's all you need to put. You cannot mention hydrolysis. Don't mention hydrolysis, you'll drop a mark easy. DNA, sorry, that's DNA helicase. I always get the names mixed up, don't I? Helicase breaks the bond. Polymerase, it joins adjacent nucleotides, or it forms the phosphodiester bond. It forms a phosphodiester bond. So that's what you need to be able to put. I talked about condensation, hydrolysis, not what it was asking for. Literally, helicase breaks the H bonds, polymerase joins the adjacent nucleotides by forming phosphodiester bonds. So yeah, a very easy mark lost there. 2 easy marks. Next, this question was very long. It was talking about explain how this virus can cause cancer, and it explained that it contains genes for proteins that cause human cells to start dividing. Literally, all it was asking for was for you to say uncontrolled cell cycle. So there you go, guys. That's 2 questions I've noticed that when it's asking you about cancer, all you need to put to get a mark is talk about uncontrolled cell cycle. This question was an evaluation where you need to give yes and no, and I would have probably gotten less than a mark for this, but I rounded to 2 because I think I mentioned 2 things. It was basically saying, should you give this vaccine to 10 to 12-year-old boys for a disease which is transmitted through sexual contact that only affects girls? And the whole idea you had to talk about here was yes, because it prevents future exposure to the disease. Yes, because it contributes to herd immunity. No, because the cancer only affects females. Things like that. And I kind of rambled there. I rambled about some stuff. The thing I've noticed, final question, is just getting some definitions right. Species richness is the amount of species in a habitat. Richness is the amount of species in habitat. Index of diversity. The index of diversity, literally all that's saying is the relationship between number of species and number of organisms in each species. I talked about it in relation to the Simpsons indexity of diversity. I talked about how you would have to divide. That's not what it was asking. It was literally saying it's the relationship between the number of species and the number of individuals in each species, which I didn't get. So that's it. There are a couple more questions here which I missed. There's one talking about observing mitosis. There is a graph question. State the name needed to determine the difference between... State the null hypothesis. Have we even done that? State the statistical test needed to determine whether the difference in the mean is significant. I think that's called the t-test, I'm pretty sure. But we've never learned that. The scientists recorded these measurements for the tissue, use this information to calculate the number of cells. So you would just multiply that by something. Okay, that seems fair enough. Two considerations to ensure the comparison is valid. Same type. Looking at the same difference between the root tip. But this is a reading question about carbohydrates, mass transport. Explain how sucrose transport proteins in leaf cells enables the production of a pressure gradient in the phloem. Sucrose transport proteins in leaf cells. Plants transport most of their carbohydrates as sucrose. The phloem has sucrose transport proteins in the membrane. Potato plants with fewer... Okay, hold on. Plants transport most of their carbohydrate. The phloem has sucrose transport proteins in the membrane. So explain how sucrose transport proteins in leaf cells enables the production of a pressure gradient. I don't know, that's worded really awfully. How do transport proteins enable the production of a pressure gradient in the phloem? Because they diffuse via facilitated diffusion out of the leaf cells. There is a lower... I don't know. That's a weird question. I don't understand that. Explain why potato plants with fewer transport produce fewer potatoes and sugars. That will obviously be because less sucrose transport means... I don't... Oh my god, I hate having to explain these. Oh my god, I'm in a really awful question about... Secondary structure is held by hydrogen bonds. Tertiary structure allows a protein to be positioned. Oh, okay, but honestly, some relatively tricky questions. So yeah, I'm happy with getting 70%. Obviously, if you do the math, if I got every other question there wrong, 35 out of 50, let's add... If I got every other question wrong, I would have still got 46%. If I got every other question in that paper wrong, and I know I probably wouldn't have, I probably could have gotten, let's say 10 more marks, then I would have gotten 60% for that paper. All that would have mean is that I would have needed, basically, in the next paper, in order to get 50%, I would have needed to get approximately 30 marks. In the second paper, I would have needed to get about 30 to 40 marks to get a grade A. If I got every other question wrong there, right? Wait, no. If I got 10 more marks, in that paper, which seems reasonable, to be fair, that means I would have gotten 10 out of 24. That means I would have needed to get, in the next paper, at least 35 marks, which I think is doable. No, sorry, 45. Hold on. Yeah, I would have needed to get approximately 40 marks. So literally in both papers, guys, you literally need to get, in both papers, about 40 marks in each paper. And I'm just going to go through and literally just comment right now. I'll make a count. How many marks is due to a lack of knowledge? How many marks is due to a genuine mistake? Lack of knowledge. Literal misreading the question. Lack of knowledge. Lack of knowledge. I guess overthinking the question. Misreading the question. Lack of knowledge. Overthinking the question. And lack of knowledge. So right there, if I made a very quick pie chart, I would say about 50% of the reason I dropped marks was due to a lack of knowledge. Like literally a lack of me not knowing the exact definitions, me not knowing the exact process, me not knowing the definition. I would have gotten maybe 8 marks just had I known that. And if I got 8 marks, I would have ended up at 85%. I would have gone up by 27% had I literally just gotten, 17%. I would have gone up by 17%. So yeah, there you go. And I think the other 25% was due to misreading, and the other was overthinking. Again, in a lot of the table-related questions, I literally lose marks for making the simplest of mistakes. Like, it could show a table and it would say, suggest how, and I wouldn't put A decreases, B increases, this suggests that they are linked. I wouldn't put that because I would overthink the question. So there you go, guys. There's a little bit of reassurance right there. 17%, let's just round it to 20. 20% of the marks I just dropped in that paper, which I've never seen before and didn't do much revision for, was due to a lack of knowledge. It wasn't due to struggling with exam technique. It wasn't due to, not understanding X, Y, and Z. I dropped 20% of that paper because I genuinely didn't know definitions. I didn't know the definition of, I didn't, what word to say? I didn't know the definition of overlapping, non-overlapping, which literally just means that the bases, what does it mean specifically? Each base only codes for one codon. Each base is only part of one codon. Right? That's what it means. Each base only part of one codon, yeah. And universal just means the same codon always codes for the same amino acid. So, what am I going to work on then, guys? What am I going to work on? Well, that's DNA related. This is also DNA related. So clearly DNA is a bit of a weak point. Lipids, literally general knowledge about lipids and the phospholipid bilayer. That made me drop a load of marks. And finally, species richness. That would have literally been 2 marks. And then this would have also been 2 marks. So overall, things I'm going to work on for the rest of today is general knowledge of DNA and lipids. I'm going to take a quick look at those definitions again later today to remember them. And yeah, that's about it. Thanks for watching.